everybody. Welcome to the next Tika talk. This is our fourth Tika talk, I believe, Carol. And,
Yeah, it's the fourth. Yeah, we started in January.
okay, um, on our April Tika talk is going to be really good because today we're traveling to Hawaii, and we're going to have two really good talks from Dr. Jason Cantley and Dr. Tim Gallagher. And the talk is being recorded today, so you can share it, on social media and tag TICAtalks, the Twitter. Also, you can sign up for TICA, the International Compositae Alliance. Now we have like an official registration, and you can join TICA and be informed of all of the things happening and get a copy of the newsletter or contribute to the newsletter and join the journal club and just stay in touch. Um, People are still filtering in, but we can go ahead and get started with our first talk today. We're going to have Dr. Tim Gallagher, who is the curator of botany at the Bishop Museum on the island of Oahu in Hawaii. So Tim, if you'd like to share your screen and take it away.
Sure thing. Are you seeing the screen?
Yeah, it looks great.
Does it say Plants of Hawaii on it?
Yep.
Hey, all right. Okay. Well, aloha, everyone. Tim Gallagher from Bishop Museum, and this is my first time at a Tika talk. I'm really excited to be here. Thank you for having me. I'm just a dabbler in the Compositae, unfortunately, not an expert like many of you are. And my talk today will touch on the Compositae as an example of what we're doing with Plants of Hawaii. And Plants of Hawaii is kind of like an e-flora for Hawaii. And just to make sure we get lots of beautiful pictures in there, this is some of the diversity, both native and non-native, that we have in Hawaii. So, and before I go any farther, I just want to acknowledge my team at the Bishop Museum. Kelsey Brock, Barb Kennedy, Pumehana, Clyde, and Nick. We have been working for the past 2 years on Plants of Hawaii, which, as I mentioned, is an e-flora, which will really bring together a lot of information about all of the plants, native and naturalized in Hawaii. And so you've probably, if you've heard talks about Hawaii before, we like to boast about how isolated Hawaii is and about our wonderful diversity. And Hawaii is really one of the most isolated places on Earth and has one of the most isolated biotas. And so you can see on the right there how how far away we are from a lot of the continental sources of the plants that have come to Hawaii. And as far as our native flora goes, um, 259 initial colonists has given rise to 1,193 of the native plants that we have today, 90% of which are endemic. And so that on average, that comes out to about 4.6 taxa per colonist. And so Hawaii is really a laboratory of Compositae evolution. There were 10 to 11 pre-human successful colonizations of Hawaii. Jason may decide to correct me on this later. Please go for it, Jason. And these are the native lineages of Compositae that we have in Hawaii. And compared to kind of the average, there's a lot more evolution happening. When these Compositae arrive, they have really undergone really spectacular species radiations. And so for each of the colonists, there's 8.7 to about 13.3 species per colonist, or taxa, depending on exactly how you're counting things. And some of the most impressive are, of course, the Silver Sword Alliance, Lipokita, which we'll hear more about, Bidens, and Tetramalopium. And I've been doing some work on Bidens, and we will soon be doing work on Tetramalopium. And just to note, there's 2 more species of Hawaiian Tetramalopium, which are currently being submitted. And so we also have a lot of non-native Compositae in Hawaii. There are at least 424 non-native taxa, At least 114 of these are considered naturalized invasive species. And each year, 1 to 2 of them are put into the category of being truly naturalized. So they're escaping cultivation and they're starting to invade native ecosystems. Right now, we have 19 potentially naturalizing species on our list. And actually, as a part of this process, we've decided to go through all of the flora, including the cultivated flora, and really assess how these things should be classified. So there's 154 that remain to be assessed. We actually note about 10 each year that potentially could become naturalizing. This year, we have 2 that are clearly in the list of naturalizing, and you can see them on the right of the screen there. So we started this project back in 2019 because there was the thought among a lot of the people working in Hawaii that there needed to be some new organization to the information that we have about the flora of Hawaii. The manual of the flowering plants of Hawaii is really our Bible, and it was our Bible for the flora. And it was written in 19. It was completed in 1990. And over the intervening 30 years, there's been a 10% increase in the number of endemic species that have been described, 86% increase in naturalized species. So the size of the flora is much greater than it was. And we don't necessarily have the keys to identify things. And so many names have changed. So it becomes very difficult for people working in native species conservation and invasive species work to really wrap their heads around what's going on and do research and do identifications. So we conducted a poll, got 116 respondents, and asked them several questions about the flora, about the resources that are out there. And they all are using a bunch of different resources for identification. The number one resource is actually a private website by a company called Star Environmental. People definitely use the manual of the flowering plants, but there was a clear response from the people working on the ground in Hawaii that there needs to be an update. So these are some of the specific requests that we got from people, and it goes on and on. These are just some of the ones that we got that they really capture the thought in Hawaii that there needed to be kind of an all-inclusive site that had keys, descriptions, lots of good photos, and other information so that people could go to one place and really get the information that they needed. Whoa, that's not supposed to look like that. Sorry. So we took all of what everyone had said and um, kind of put it into this organization chart here. We took all of the resources that were available and out there and tried to map it out and see what we could do. And so if we start down on these blue squares on the bottom, there's names in lots of places. There's all sorts of checklists. There's cultivated plant checklists, ethnobotanical checklists. There was the Manual of the Flowering Plants of Hawaii and the accompanying website. The Bishop Museum had its own checklist, which had slightly different taxa in it. There's the USDA, there's the federal government, there's the IUCN. And so we wanted to get all of this into one place. So we took all of this in and are still cleaning this up and into one taxon table. Then we looked at occurrences and images, and traditionally herbaria look at specimens, but now there is so much more information out there. There is survey data that's publicly available. There's images through iNaturalist. And so we wanted to really take all of that in as potential evidence. And we wanted to build that into Plants of Hawaii as well. And so that ended up being 3 tables: an images table, occurrences table, and locality table. There's all sorts of research on plants, of course. It's what we do. And so the manual, specimens that we have, all of that ends up into our measurements or facts table. And really what we are trying to do, and this is a work in progress, so it's not complete, is build all of this into one place called Plants of Hawaii. Ooh. And in the process, we hope to reduce some of the redundancy. So we're getting rid of some of these databases that we previously hosted and putting it all into the one spot. And so, as I mentioned, this is a work in progress. And so one of the things that was helpful for us in doing this work was to build ourselves a dashboard. And so this is the Plants of Hawaii dashboard, which is currently really only available to the people working on it. And so a lot of us are familiar with dashboards now. You can kind of click on any of the graphical items and get the underlying data or drill into more detailed data. So when we got all the names together, we ended up with 86, almost 87,000 names. And we've gone through the process of trying to classify them to determine if they are accepted names, synonyms. You can see over here, we still have some that we are reviewing. Unfortunately, a lot of people misspell names, especially the USDA. If there's anybody with the USDA here, really sorry, but there's a lot of spelling errors. And so we ended up with over 1,000 name variants, the vast majority of those USDA spelling errors. And then we decided to look at occurrences, and the Bishop Museum has 171,000 plant specimens from Hawaii, by far the largest anywhere in terms of actual specimens. But there are aggregator sites like GBIF and iDigBio, and these have so many other specimens from other places. And we came to the realization that we can't just rely on what we have, because that's only part of the story. So we've started to build mechanisms to ingest all of these data from all of these other sources into our database so that we can compare them to what we have. In some cases, we can find determinations that were made on other specimens that are duplicates of specimens that we have. And we are in the process of building this pipeline. And our goal is to ingest 500,000 Hawaii plant distribution records and create a bioinformatics system to keep this database connected to all these other database sources. And so this is kind of the model that we are using. So the Plants of Hawaii will attempt to do all of these things. So we will be collecting information from all of these sources, cleaning and normalizing it, and then merging and extending. So often there are duplicates, there are specimens that were collected from the same locality. We want to be able to link these as an extended specimen network, visualize these, especially geographically over the landscape, and then be able to share this data with anybody that wants to use it. One of the big challenges that we've had is classification of plants. We can easily tell if something is native or non-native, and we pretty much know if it's endemic or indigenous. But it becomes difficult once you classify things as non-native. Are they only in cultivation? Are they found occasionally outside of cultivation, potentially naturalizing, or truly naturalized? And so Kelsey Brock, who works with us at the museum, published this paper that you can see at the bottom here, which gives specific guidelines for Hawaii on how to classify these things into the various categories that we have here. And we're in the process of doing that. And so this is another view of our dashboard. And in this case, all taxa, these are the accepted terminal taxa only. And of all of these, we have 1,170 that are endemic. There's actually a few more that are still in this no status zone here that need to be addressed. And then all of the other classifications that we have, including the 1,999 that are yet to be classified. This is the Compositae. And so we also have a bunch that have not been classified. Most of those are cultivated, but we need to know about them because that's where we get our invasive species from mostly from cultivated plants that start, for whatever reason, to escape into native systems. 130 plus a few more are endemic, the one indigenous Asteraceae, and a lot that are cultivated and naturalized. And so the great thing about these dashboards is that you can slice the data in many different ways. You can slice it by family, by phylum, by classification system, and it really helps to get a good good idea for what we have once we can slice and dice this way. One application of this is for the noxious weed people that want to know where noxious weeds are. And so we were asked by the Hawaii Invasive Species Council and the Noxious Weeds Working Group to use this data to build a noxious weed distribution map, which is an interactive map on our dashboard. Unfortunately, we found that only about 17% of all the Hawaii plant specimens, that includes the Bishop Museum and all the specimens from around the world that we've been able to ingest, only 17% have coordinates, and the vast majority of those coordinates are not very good. Very often they put the spot in the middle of an island because the location said Oahu, so they found the center of Oahu. One other thing that we've been doing in parallel and part of this project is building a massive lucid key. And we started with the Asteraceae, and we hope, sorry, Compositae, tried not to do that, which Compositae, and we've been going through all of our specimens and published descriptions and adding in characters. And so we've used a very comprehensive set of characters, 300 characters and over 2,000 character states, because we are using this to also build descriptions. And then we're going to extract some of these characters to put into our character database so that people that come to our site can do specific queries, like where are all the yellow flowered plants, or what is the distribution of woody plants, anything like that. So we are in the process of doing this. We have preliminary data in for most of the Asteraceae at this point, native and naturalized, and a few others that we want to keep track of, especially potential invasive species. And over the rest of this year, we hope to be able to finish this and make this lucid key available online. And Jason Cantley's also working on this project with us, and his student is as well. We're very, very grateful for that. All of what I've been just talking about will be, and is to some extent, available on Plants of Hawaii. And so this is patterned very much after Plants of the World Online, which I'm sure some of you are familiar with. And basically anyone can search by any word that describes a plant or a plant name or a common name or a geographic locality on the front page here. And they would get results like this in individual tiles, and then they can click on any one of the results, which are color coded by native or non-native, and they come to a species page. And you'll see that this particular silver sword here, it has some images in it, but a lot of the description information, a lot of the other information is not yet on the site. And so we have our team as well as our volunteers working on this, and we hope soon to be able to populate all of our description data into the database. And actually Asteraceae will be first, as I mentioned. Compositae will be first, as I mentioned. And we have information that we are accumulating from in all of these sources. So synonyms, accepted subtaxa, uses and culture, information about propagation, where the species are located on each island, as well as all of the different conservation statuses and some natural history information. One other tool that I want to just briefly mention is our checklist builder. And so all of the character data can be used to do a kind of a broad global search across Hawaii for different things. So we can look for plants that are just indigenous or those that have certain ecological characteristics. This is where we also hope to put some of our morphological anatomical data, such as flower color or growth form, and so that we can do a visual map-based search for those characters and find out where these co-occur. We hope that this will help with studies of functional traits and things along that nature, and also help with identification. So if someone sees a red flowered thing on a specific island, they can pick the island, pick red flowers, and get to at least the list of things that are known to exist there. So I'm really sorry that this talk didn't include more composite and more pictures and more interesting things, but that's okay because Jason's talk is coming up next and he will have some really interesting things to say about that. But I do thank you very much for listening. I will stop sharing.
Tim?
Yeah.
Tim?
Yes.
Randy Bayer here. Are you going to put the Lucid key? Will it be web-based?
It will be web-based, yes.
Uh-huh.
Yeah, we hope to have that done by the end of the year.
Have you tried to build a key yet? Or you just
Yes,
put it in?
we've been working on it for about 2 years now.
Does the key work fairly well?
That remains to be seen,
Okay.
truthfully. Yeah.
Right, yeah. You say you have 3,000 characters? Was that my understanding?
We have 200 characters, true characters. There are a few others in there that are
200.
not true characters, but then a lot of character states.
Oh, I see. Right. Okay.
Yeah.
Yeah, that's good. Yeah.
We're going to go through. So what we did initially is we took the manual of the flowering plants of Hawaii, and we've been going through and just putting the characters in as written. And we
Right.
immediately realized that people describe things in very different ways, use different terms. And
Right.
so our next phase of this would be merging characters, because sometimes if we have someone that just doing data entry, like a student, they may not realize that two things are really the same thing.
Right.
So we
right.
just have them put it in as written.
Right. Do you have a list of those characters that you could share with me?
Yes. Yes.
I've been
Yes.
trying to put together a key for Antonaria. I've started it and worked on and off on it, but I worked on a lot of those keys when I worked in Australia with Cyro,
Oh,
the
sure.
eucalyptus key. So I'm very familiar with Lucid and its development. But
Yeah.
I
I could
wouldn't
definitely
mind seeing the list of characters that you've used.
Sure. Yes, there will be some funny things in there, I'm sure, but because
okay.
we're still cleaning. But yeah, I'm happy to share that.
Be curious to see what you've got. Yeah, if you would, please. Thanks.
Yes, sure.
We got one question in the chat. Daniel Park doesn't have a mic right now, so he's asking if you have explored using the BIEN database and associated tools, for example, TNRS, for scrubbing standardizing the data.
I have not. But I would love to learn more about that. That's scrubbing standardizing the data is kind of the bane of our existence right now. So yes, I should definitely learn more. Does anyone else have experience with that they can share? Many, yeah. So BN goes through many of the same processes. Yeah, I definitely want to learn more. There are tools out there, and sometimes it is hard to find these tools and know about them. So I appreciate that.
Okay, unless there's any more questions for now, we can move on to the next talk, and then afterwards we might have some extra time. So if people want to have kind of like a more open discussion or ask Tim about his ongoing research on tetramalopium and other compositae in Hawaii, they can do that. And but first, we're going to have another great talk by Dr. Jason Cantley, who is the herbarium curator and assistant professor of botany at San Francisco State University in California. And he's going to talk about Compositae pacifico. So take it away, Jason.
Okay, so I'm really excited to talk about this. Pacific plants have been sort of something that's been on my mind since I started my PhD at the University of Hawaii at Manoa. And Tim Gallagher and I were graduate students in the same lab together. So it's really nice to have us talking together on stuff that is really we're both passionate about. And today I'm going to be talking about patterns of composites in the Pacific, and then we'll dive a little bit more deeply into a lineage I'm starting work on, the Wollastonea and the Paquita lineage in the Pacific. And this study really is part of a much, much larger project that's been an ongoing source of research for many, many years. And it involves many different peoples from many different kinds of organisms across the Pacific. But today I want to pay special recognition to Vicki Funk, who I was lucky enough to work with directly in our lab in the early 2010s, while she was a visiting scientist in the lab we were working in. And Vicki really has been interested for a long time in sort of the evolution of life in Pacific Oceania. And she wanted to know what are the relationships among the species that occur there? Are there any general patterns that explain the distribution of clades across the Pacific? And how have these taxa arrived to, and then in some cases, subsequently diversified across the Pacific? And this is a really interesting thing. And Vicki actually left us with a sort of unfinished manuscript regarding the Pacific composites today. So if you sort of put that in your head and have some ideas about how we can continue forward with this project and the data that I'll share, I would love to have your thoughts. Oops, change. So many of you may not know about the distribution of habitats in the Pacific. And the Pacific is really an ideal location to test many evolutionary hypotheses for several different reasons. First of all, there's a lot of endemism. The land masses across the Pacific are generally very isolated, and they usually have never been connected to a continental landmass. These islands are generally small, they're young, and they have dynamic landscapes that often host heterogeneous landscapes with many different habitat types. For example, the Hawaiian Islands has every type of habitat type except for taiga and tundra. Gene flow as a result of this isolation is often really limited from continental source populations. And overall, the total diversity on island ecosystems is much less than continents. And because of this, these are simpler systems with species to species interactions, and it makes it a bit easier to isolate variables and test evolutionary hypotheses. And to orient you a bit in the Pacific, it's really, really huge. The Pacific Ocean covers 2/3 of the surface of our planet, and it has over 20,000 islands that are habitable by terrestrial organisms. I mean, those numbers just blow my mind every time I share them. And thinking about how organisms can arrive to these islands, you can use the mnemonic to remember they can arrive with the 3 W's: on wind, by water, or in the wings of birds. And whether or not a species will land to a certain location in the island, it really does plays with this degree of isolation an island may have or the size of those locations. And the yellow here is Polynesia, and North Polynesia is usually considered just the Hawaiian Islands. And then these regions down here is sort of the Southeast Polynesian Islands. New Zealand often being part of, culturally is part of Polynesia as well. And then we have sort of Australia and Melanesia, and then Micronesia, which are much lower islands in general over here before we reach Asia and Southeast Asia. And the Pacific is, as I mentioned, gigantic, and the distances from continental sources are immense. So take, for example, the width of the North American continent at roughly 4,500 kilometers. That's comparable to, say, the distance of the Marquesas Islands to the coast of Mexico, or Hawaii to around San Diego, the closest continental land mass. These are really, really remarkable distances. And when you start to think about it, it's amazing that plants have been spreading across our planet in the fashion that they have. A lot of the work looking at the origins of taxa in the Pacific had been thought about for a long time, but I wanted to point out this sort of pivotal moment when Vicky Funk and Warren Wagner detailed sort of some biogeographic hypotheses about of taxa that occur on the Hawaiian archipelago. And this work definitely incorporated established concepts of insular plant evolution, such as long distance dispersal events that were required to land on isolated land masses that were never connected to continental sources. They also detailed this idea of a progression rule where a colonist to Hawaii would likely arrive on the oldest emergent high island. And then as younger islands were generated on the Pacific plate, the species would disperse down the island chain in a progression-like fashion as habitat became suitable for their colonization. The result of this, of course, has led in the Hawaiian Islands to many adaptive radiations. And there are really cool evolutionary concepts such as the loss of fruit or seed dispersibility or development of traits that kind of fit this mold of something called island syndrome. And the island syndrome is a little bit more well known for animals, but in the case of plants, there are all kinds of things such as an increased woodiness from an herbaceous colonist, and this could be kind of like gigantism. And several plants that are larger on continents actually do become much smaller and diminutive in form. And in this image here, I wanted to make sure I pointed out the classic case of loss of the seed dispersibility or akene dispersibility for the Hawaiian lineage Bidens. The colonist who had akenes very similar to, or it's postulated, similar to this Bidens pilosa, where you can see these barbed pappus awns here would be stuck in perhaps a bird's feather and then dispersed to the Hawaiian Islands. But upon subsequent cladogenesis of the lineage in Hawaii, these dispersal mechanisms have been lost and the seed morphology has changed to reduce dispersibility away from the Hawaiian Islands themselves. So Vicky, really being Vicky, had always had in mind, I want to study comps in the Pacific. What are those patterns of compositae in the Pacific? And she asked a lot of them of similar questions, like how many species and genera are there endemic and indigenous across the whole Pacific? Where have they radiated? How have they radiated? How old are those lineages? And how did these lineage disperse? Did they just disperse to an island archipelago and stay there, or was there exchange among different archipelagos? And we calculated, Sterling Keeley, my graduate advisor, and Vicky Funk and myself did some calculations, and we came up with 174 Pacific compositae that are total across the entire Pacific, and not including New Zealand, which we left out of this analysis. And what we find are some really interesting patterns. And concerning the continental source of a lineage to an archipelago in the Pacific, we actually see the most number of introductions come from Australia and New Guinea, and then secondly, from Asia and Southeast Asia. But we can also see that the generic diversity really by far comes from lineages from from Australia and New Guinea region. But we have a really interesting pattern concerning the number of endemic species. Here you can see in gray, for both the number of species and the number of endemic species, the amount of diversity with a North American origin is really described by two spectacular radiations, Bidens and the Silver Sword Alliance in Hawaii. And you can further sort of fuss with these data and break them, the colonizing continental source, down by archipelago. And when you look at this, you see that the Hawaiian Islands actually have colonizers from 4 different continental areas, the numbers in this bar actually indicating the number of introductions. And this is perhaps not surprising, considering Hawaii is the highest and the largest in terms of landmass habitat of in the Pacific. And these other archipelagos are much smaller and have less habitat heterogeneity. So it's perhaps not as surprising that they have colonists from less number of continental sources. If we consider just the endemic species of Compositae alone, we can see that over, and there's 158 endemic species. The rest are about 20 indigenous. The 6 radiations represent 144 of 158 endemic species, and that's 91% of all of that diversity. And remarkably, out of that, Biden's represents 1/3 of all Pacific Compositae diversity. That's incredible. And you can see that here in these. If you add together the radiation of Biden's in Hawaii and the radiation of Biden's in Southeast Polynesia, you get roughly 1/3 of this pie. All large radiations in the Pacific of Compositae occur in either Southeast Polynesia with Biden's or Aperanthus, or in the Hawaiian Islands with Biden's Lipikita wollastonia, Tetrabolopium, or the Silver Sword Alliance. And one thing that we haven't touched on was sort of the geography and the geologic history of islands in the Pacific. And if you think about on a geologic scale, islands are very ephemeral. They're not around for very long in many cases. And in the case of the Hawaiian Islands, the oldest emergent high island that had diverse habitats is thought to be Kauai and has been emergent since about 5.2 million years ago. And we see that the radiations of Compositae in Hawaii actually are younger than this age. And that actually is really cool. It's a pattern that's reflected across many organisms in the Pacific or in the Hawaiian archipelago. These studies that have done ages on Hawaii have also included other taxa from elsewhere in the Pacific. And as a proxy, the lineages of Lipikita, Tetrabolopium, Biden's elsewhere in the Pacific are also young in age. So I wanted to take a moment to actually show you what these lineages look like. You might know the charismatic megaflora adaptive radiation of the Silver Sword Alliance. These are a spectacular radiation of about 37 species that have diversified into 3 distinct genera. And they have arrived to Hawaii roughly 5 million years ago from a single colonist that's related to the currently extant California tar reeds of tribe Matii. And you can see here, these 3 genera are represented. We have our Gyrosiphium, literally translates to silver sword in Hawaii, having these spectacular adaptations to life in subalpine habitats. They have triangular leaves that have anti-freeze properties that prevent them from freezing. We have dry forest endemic Wilksia on the island of Kauai that's more arborescent. And then there's this genus with a huge amount of ecological diversity, such as a bog cushion plant, Dabaudia aleale, or colonizer primary land on new lava floats with Dabaudia scabra, and even there are rainforest liana species in this genus. Really, really textbook, remarkable adaptive radiation. Biden's in the Pacific is also remarkable. Not only is it one of the fastest documented radiating lineages in the world, it also is the largest radiation in the entire Pacific. Of the 152 species in this genus, 41 of them exhibit the greatest ecomorphological variation of the genus, and they all occur in the Pacific. And you can see here in this phylogeny, many of you on this call might actually be authors on this paper, so thank you for this really incredible work. This whole upper group here, in seafoam green, I guess, is Hawaii, and this group down here in the blues are from Southeast Polynesia. So there's just been a remarkable radiation in 2 different areas in the Pacific. And one species that I'll call out is this spectacular evolutionary shift from being pollinated by insects to bird pollination in the Hawaiian endemic species Biden's cosmoides. You can see that this this inflorescence has changed as conformation and become pendulous and a little bit more deep, which helps facilitate birds doing their pollination in this species. And lastly, the last really large radiation in Hawaii that I want to talk about more is the Pacific Wollastonea and Lipakita. There is a widespread species, Wollastonea biflora, that inhabits coastal strand or beach-like habitats across many islands of the Pacific. But in Hawaii, there are strand species as well as species that have diversified into many habitats at higher elevation. And it's thought that the origin of these that came from Southeast Asia and Australia or New Guinea has arrived to across the Pacific by water dispersal. Okay, get that, Compositae arriving by water dispersal. That's wild already. And now there's actually some questions of whether or not Hawaii Lipakita is an adaptive radiation. And I'll talk about that more in just a moment. So to summarize, this sort of unfinished manuscript that we were working with Vicky Funk before she passed, we actually have some really cool synthetic statements that we can say about Pacific composites. There's 174 taxa, 6 of the lineages account for most of the diversity there, and many of them are endemic, 154, rather than being indigenous. Most of them have undergone some sort of cladogenesis, and the biggest radiations occur in Hawaii or Southeast Polynesia. We also know that they are primarily young in age, and dispersal is going not only from continents to sink archipelago, but also to and from archipelagos, such as the Hawaii to the Marquesas Island and in the reverse. Okay, so I'm gonna shift gears here and we're gonna slide into a little story about the Pacific Wollastonea and Lipakita, which is research that I started with Vicky while she was visiting in Hawaii, but I'm continuing now as an assistant professor at San Francisco State. And the lineage of Lipakita has perhaps been the least studied, or it's the last to be studied of the adaptive radiations in Hawaii. And the reason for this is because there hadn't been a really great phylogeny produced for the relationships of genera in the Ecliptini. And Ecliptini is challenging because it is global in its distribution. And of the genera in something called the Melanthera Alliance, there are a number of genera that are found all over the world, and you can see them here. And this distribution confounds us understanding the origins of Hawaiian and Pacific taxa. So we have Wollastonea, that's in Southeast Asia, New Caledonia, and then actually widespread one species from Africa, Asia, Australia, the Indian Ocean, and Pacific Islands. There's a radiation of Lipotrichi in Central Africa, a sole species in South America on the eastern coast, Echinocephalum. Southeast United States and Central America have white-flowered Melantheras. Australia has a radiation of 8 inland xeric-adapted Apowallastonias. And then, of course, in Hawaii, there's an adaptive radiation of around 20 taxa. And we did recently, in 2018, with Bort Edwards as the lead author in this paper, publish a phylogeny from this work. And what we found is, if you look in the phylogeny, the blue taxa here represent Wollastonea, and the green are Lipakita. We found that they are sister to each other. Sorry, Lipakita from Hawaii and the Wollastonea from Pacific and Asian locations. They're sister to each other, and they, taken together, are sister to the African taxa Apowallastonea. We further understand that the Hawaiian taxa, several of them may be polyphyletic. This was based on just a few gene regions, ITS, ETS, and maybe a few chloroplast regions. So we don't, and because of that, these polyphyletic lineages in Hawaii need closer investigation, and there's also not much in the way of support among these taxa, so poor resolution. You'll also note that there's a darker clade here and darker green, and this is a separate section of tetraploid taxa in this lineage. And I'll talk about that, the implications of that in just a moment. So that polyploid lineage is actually a whole separate section Lipoceta, and they have different morphology. Primarily, you can notice the 4 merис flowers and stems that are rounded or weakly ribbed rather than strongly ribbed, which is known for the diploid taxa of Lipoceta in section Aphanopappus. And what's interesting is this is an allopolyploid origin. The chromosome numbers don't link up exactly correctly, and some studies from the 80s and 90s really have demonstrated that this is not an autopolyploid development. And so what we have here is actually a ghost taxon that's living within the Hawaiian Lipoceta, and we actually have no idea where it comes from. A lot of the texts say it's like some sort of Wedelia, which is a really challenging Ecliptinae genus that itself is polyphyletic. And this leads me to sort of the last question and sort of current research in our lab. And we're really interested in understanding whether or not Lipoceta in Hawaii is indeed an adaptive radiation. And there are all kind of qualifiers and different definitions of what really qualifies a lineage as an adaptive radiation. But what I'm mostly interested in is the remarkable evolution of this lineage. Just by quick glance of a little more than half of the species here, you can notice that they occur in many different kinds of habitats, although it's limited and not as remarkably diversified as, say, the Hawaiian silver sword alliance. We have taxa that live on the coast, like Lipoceta integrifolia, that has succulent leaves and could be, in fact, a halophyte. We have large palmately trilobed leaves of Lipoceta rockii. There are species that are an annual. Lipoceta rimii is an annual, and that is not very common in island floras at all. We have, and then a few other things that I'll call out are this really crazy leaf variation in the genus. We do have some interesting ecological developments such as coastlines, rocky cliffs, stream beds of mesic forests, and dry coastal shrublands. And really, to answer evolutionary questions in Lipoceta and Wollastonea, we need to produce a more resolved phylogeny. And with that phylogeny, we'll be able to ask questions of whether or not Lipoceta follows the progression rule. Remember, that was postulated by Funk and Wagner in their book in 1995, of species landing on an oldest emergent island and then dispersing downwards to successively younger islands. We also want to know whether or not the ancestor to the Hawaiian Islands arrived via hydrochory or water dispersal. And I'll show this here, and this links in really well with the third question of, does Lipoceta have a loss of dispersibility as it evolved into different habitats in the Hawaiian Islands? And here we have a Hawaiian endemic, Lipoceta integrifolia, found in coastal habitats, rocky areas, and coastal strand. And its seeds, like its progenitor species, Wollastonea biflora, actually do float in salt water and remain viable too. We see here, if you do a cross section of the seed, there is this area that's been described in the literature as corky. And this is the outer layer of the achene, and it has a lot of air bubbles. It kind of looks like, if you look at honeycomb candy up close, it's just full of these little air bubbles. And this is actually now a buoyant tissue that keeps these seeds floating on the surface of water. And that, I think, is the mechanism for how these species are distributing across the Pacific. And so what we are really interested in is looking at, in much greater detail, how, first, what are sort of the early diverging Lipikita species, and what is the seed morphology of species that occur in other habitats in Hawaii? Do they retain this layer of tissue, or have they lost it subsequently? And so that leads me to sort of the current thing where we're at in the lab. My new graduate student, Jennifer Branson, is already starting to extract DNA, and we're going to be sequencing this summer for a hide-seq phylogeny using the MyBates Compositae specific genes for over 1,000 genes. And with that, we'll be able to look into the biogeography of how the species are related to each other. We'll be able to assess the polyphyletic lineages, and especially we want to look at the sort of morphological adaptations or changes of leaves, because they seem like there's a remarkable story there as they diversify into different habitats. And that's also true for the seed morphology. We want to know a little bit more. Can we sort of piece together some sort of synthetic evolutionary story about the loss of dispersibility in these taxa? And then finally, and I don't quite know where this will go yet, but I want to think more deeply about this tetraploid taxa of the section Lepikita and that unknown ghost taxon that exists within it. What is it? What do we do? How do we discover this ghost taxon's identity and origins? And so with that, I would just like to say mahalo nui loa, or thank you very much in Hawaiian, to a lot of different people, the Smithsonian, University of Hawaii at Manoa, and of course, I have to thank field botanists and herbaria for sort of really being committed to preserving and protecting resources for these plants so that they can actually remain in perpetuity in Hawaii long after humans quit messing up all of their environments. And I'll end it with a thanks to Vicki Funk for the inspiration, encouragement, and really the sort of conversations we had that were so engaging about really Pacific comps and their spectacular evolution. So thank you. And I'll be happy to take any questions.
Great, Jason, if you want to end your screen share. Anybody who has questions for Jason is welcome to unmute themselves and ask them. But also, we have a good amount of time today. So if anybody who does research in areas that are similar on Compositae on islands and other island systems and wants to add their 2 cents to the conversation, is welcome to do so. And you're welcome to turn on your video and join the conversation.
I'll ask a question, Jason.
Sure.
The Tetramelopium that's in New Guinea, is it just one species, or are there several species in New Guinea? And where are they found? Are they in the high mountains there?
I believe there's more than one taxon, but I will defer to Tim because he's been a little bit more involved with the non-Hawaiian Tetramelopians recently. Do you have an idea, Tim?
Yeah, there is more than one. But I believe it's about a handful, 3 or 5 or 6, around that number of taxa. I know that they are in the mountains, but I'm not sure the total breadth of the habitats that they are in. But yes, high elevation typically.
For them, yeah, right.
Yeah.
And we didn't quite mention, but Tim and I and Matt Nope at the University of Hawaii at Manoa are working on Tetramelopian phylogenetics. So we have a little bit idea of how they're all related across the Pacific.
Have you been working at all with Tim Lowry?
We have tried to
Oh,
get
he's
in
retired
touch.
now. I know he's retired now.
Okay, yeah, we've been trying for a very long time actually to get in touch with him. So if you have an in with him and you want to let him know that we are trying to get in touch with him, we would really like to work with him.
Oh, really? Well,
yeah.
I heard he moved to Arizona from New Mexico after he retired. That's all I.
We may just have the wrong.
I have corresponded with him not too long ago. He's around. I will I'll write him an email and let him know you're trying to get in touch with him.
Thank you. Would
Thanks a
appreciate
lot.
that.
Yeah, he really did that classic work, you know, back in the 80s, yeah, 80s, tetramenlopium with Bob Orndorff.
I had a question.
With your Lipochetas, Jason, would you think about growing those in a common garden type of experiment to see how plastic that leaf morphology is?
I think that would be a wonderful study. There are some challenges to that. One, I'm physically not in Hawaii any longer. And two, a number of these taxa actually are quite endangered. And so
Yeah,
the
right.
permitting issue around getting enough seed source to grow for that purpose is questionable. And I actually question the ethics of whether or not I'd like to do that. But some of the taxa, especially the polyploid ones, in my experience in the field, there is quite a lot of plasticity. But the diploid ones seem to be a little bit more uniform across populations. And I guess that's to be suspected.
That's typical of what you find with polyploid complexes.
You know, I was putting together this talk, and I actually think that there might be more diversity or observable diversity in leaf morphology for the diploids. And so I think that's really intriguing to me, but maybe the plasticity and the tetraploid taxa allow them to sort of grow their leaves to an optimal level in the environment that they're in. But the other lineages, perhaps, I don't know. I'm just actually, I should just stop because I'm just putting out hand wavy hypotheses now.
All right. That's fine. Thanks.
Mhm.
Jason, if you do find that ghost taxon, and it does appear to be something that is far distant from Lipocheta, what are you going to do about the name of the tetraploid lineage? That's the big question from everyone in Hawaii, where everyone's on pins and needles about that. For some reason, I don't know. Names change. Who cares? But
Yeah.
they are interested.
People are interested in the names of these taxa and whether or not Lipocheta is considered an endemic radiation, whether out the diploids should be Wollastonia, since that's likely their progenitor. I don't know what I would do, to be honest. I'm actually comfortable with the diploids being transferred to Wollastonia and the tetraploids becoming Lipocheta. And I think that there's a lot of there's a great argument there for the morphology that matches with the diploid Lipochetas. The ghost taxon, though, you know, my mind is really reeling about how can we, without doing genome analyses for the whole Ecliptony, how can we sort of find this taxon? And also, what would be the purpose of finding that taxon too? It's really interesting, but I'm not sure what we would do with those data too. Does anybody have any insights of how we might be able to sort of search for a ghost?
Well, personally, I think we have to start with morphology.
Mm-hmm.
and look at possibilities and go from there.
Yeah. Yeah, I think that would be really great. I think
probably some chromosome counting is going to be in the mix too.
Yeah, the chromosome counting has been done extensively, and there's been a little bit of morphological assessment, but those were done roughly 20 years ago, and there was no insight into sort of the global relationships of all of the Melanthera allied genera in the Ecliptony. So I think maybe we have a little bit more direction of being able to say, maybe we should check the Central American species out.
Right, yeah. Yeah, you're gonna have to look at all possibilities, I guess, but yeah, it could be Central America. South America, you don't know.
Jason, in the broader, study of the ecliptony, were there taxa that had the chromosome number that corresponded to like the tetraploid minus the diploid number?
I should look at that actually. I don't know. Tim, do you recall?
I do not, sorry.
Okay. You're right. That would obviously be the place. If it was there, I think that Vicky would have probably more prominently put that as like, this is probably what it is in the conversations we had. But I think one of the problems is the big genus Wooddelia, and it's sort of a taxonomic lumping ground that's clearly polyphyletic. And so I think a lot of chromosome studies would need to be performed for that genus to get a better idea.
I had a separate question for you, Jason and Tim, which was, what about these lineages that haven't had really big radiations, like Hesperomania and Adenostemma, that are kind of like, there's one species and it's maybe really restricted or pretty widespread, but it hasn't undergone any kind of adaptive radiation?
Yeah, I don't have any insight as to why that would be, but I think it's definitely worthy of investigation. It's very interesting that there's just the one for Adenostemma, just the one indigenous species, and that's it. That's the only Compositae that's done that in Hawaii. Hesperomania with 4 species, maybe 3, depending on who you ask. That seems like it could be either age or that it's something about it that I do not know. Jason?
Yeah, Adenostemma was the only one that is just a single taxon. All other Hawaiian comps have undergone at least a minor degree of cladogenesis, and most of them have like 3 to 6 taxa. And I was thinking about the patterns, and it doesn't seem, in terms of the habitats that they occupy, there doesn't seem to be shifts out of their initial, of their like habitat. It just, the speciation seems to occur when they become genetically isolated on different islands. So Hesperomania is sort of one on Kauai, one in the Waianae mountain ranges of Oahu, one on the Ko'olau mountain ranges of Oahu, and then one in Maui, more or less. So that is also an interesting pattern. And the question becomes, why have they not radiated?
So potentially a dispersal issue. They may not be dispersing well or too well. I don't know. The other thing is there is always the possibility with Hawaiian taxa that they were there and they went extinct. And so possibilities.
Hello, everybody. I'm Florencia. I'm from Argentina. So I'm sorry about my English. I'm working in Adenostemma right now. I'm a PhD student, and I'm working in La Plata Museum. I'm actually doing my PhD research in morphology, systematics, phylogeny, and biogeography of Adenostemma. I'm studying the neotropical species because the genus is big and I can't afford traveling around the world. And it's very widespread in the tropics. And tomorrow I'm having my first experiment working with birds, because Adenostemma, everybody says that the dispersal is by birds, but we don't have any like facts. We don't have facts. So I'm doing my experiment with that. And it's also a question for me, why it's not so radiated in the islands. because I have some species that are in the continent and on the islands, but they are not diversified on the islands. So that's a question for me too. I'm not going to work deeply with the old world species, but I'm going to try to answer some of these questions. So it's very, the talks were very informative for me. Thanks.
Good luck with that research. If the Bishop Museum can help at all, please let me know with samples or anything.
Thanks. That would be great.
Yeah, what a great lineage to research. Good luck. Hawaiian adenostemma, is interesting. I have so little experience with it. I've seen it just a few times actually, and it is anomalous because it's the only one that's just one species of composite in the Hawaiian flora.
For what I have seen about the species, the morphology, the leaf morphology, and the flower morphology, the capital, it's very, how do you say, it doesn't change a lot. There are some postrate plants and some erectum, and there are some of them that are 2 meters tall, and then you have little plants, but the morphology is not so different. And here in Argentina, I live in Buenos Aires, and there are some plants in the river area, the river coast. Then I have a space between the north of the country and the center of the country where are no registers. So I'm trying to find out why, if it's a dispersal reason, or maybe, I mean, there are a lot of botanists that are working in that area and they haven't seen the plant. So it's a mystery for me too. So.
Cool.
Mysteries are fun.
Yeah.
That's why we do what we do. Right?
Does anybody else have any other questions or would like to join, add to the conversation? I'll stop the recording now, but we'll leave the Zoom open in case people want to keep discussing. Thank you.
Thank you for having us speak. It was really fun today.
Really
yes,
good.
thank you very much.
Thanks very much. That was very informative.